I'm honored to give you an update and a company overview on our progress to date with DiscGenics. We are a clinical stage biotechnology company developing cell-based therapies, regenerative therapies that alleviate pain and restore function to patients with degenerative diseases of the spine. We're privately held, 82 accredited investors and one institutional investor, the Mitsubishi Bank. We were founded in 2007. We moved to Salt Lake City in 2012 to commence our preclinical development. And now today in clinical stage, we have a tremendous management team that starts with Bob Wintelak, our Chief Operating Officer, and Kevin Foley, our Chief Medical Officer, recently awarded the Cushing Award by the American Association of Neurological Surgeons for technical innovation in the spine. And we have rounded out a very good advisory team of renowned neurosurgeons and spine specialists. We have currently 22, soon to be 24 employees based in Salt Lake City. We've doubled the number of employees this past year as we move to our scale up and scale out manufacturing and cold chain logistics. Our first product candidate is IDCT, which is, stands for Injectable Disc Cell Therapy. We are currently conducting a 60-patient clinical trial here in the U.S. and commencing a 38-patient trial in Japan. We're really excited about our progress against this disease, which is pervasive and affects the quarter of the population at some time or another. It's the biggest reason for disability in the world. And if you watch this progression, especially in its mild to moderate state, it is a major cause of back pain. It has a very complex etiology of inflammation and extracellular breakdown, and it really is void of a curative treatment option, which we hope we can provide. So if you look at this disease progression and the treatment options, especially in the mild to moderate space, you're going to NSAIDs, opioids, and other pain relievers, steroid injections, physical therapy, ablation, chiropractic, and other types of alternative care. And as you enter the moderate stages where the pain increases, you're really just buying time. You're maybe in some cases you will regress and feel better, but really the rock just rolls down the hill, and you really go back to these mid-stage treatments. Obviously, if it continues to get worse, surgical intervention is really the only alternative, and quite frankly, that surgical intervention, especially infusion, works about 50 to 60% of the time. So this mild to moderate space is 7 to 9 times larger than the fusion space. And once fusion surgery takes place, you're locking down those vertebral bodies to get the pain off where those degenerative discs were. And then at some point down the road, as you lock down those vertebral bodies, you create adjacent level disease on the bodies that and the discs that are continuing to work. So it's a very pervasive disease, and we've been working on a solution here for quite some time. If you look at, and really our solution centers around discogenic cells, and these cells are therapeutic progenitor cells of a homogeneous population. They're derived from adult donor disc tissue in a single lot, and they are allogeneic yet homologous. And so they come from the disc, and we run them through a culture system to go back in and treat the disc. And we've done this through extensive preclinical data, and we have a dual hypothesized actions of regeneration and an anti-inflammatory aspect. As you can see through this mixed leukocyte reaction we show on the far left, and the fact that the cells produce extracellular matrix around aggrecan and collagen II, which is what 98% of the nucleus pulposus consist of. These cells are also very robust through cryopreservation, and we do this in a two-step process in our manufacturing. And this process, we transform these donated disc cells into these therapeutic progenitor cells. We do this without gene editing or induction. This is protected by 24 issued patents with 15 pending. We also have a lot of trade secret protection around the manufacturing process. This is an allogeneic model. A single donor lot produces thousands of doses, and it is off the shelf in a cold chain logistics from our freezer after the manufacturing process, hopefully to the freezer in the clinic. This is a very reproducible process of a very homogeneous cell population of over 20 donors to date, subject to potency, identity, purity, and intense safety testing. So our first product candidate is injectable disc cell therapy. This is a very cost-effective treatment compared to what is essentially ineffective surgical care and eventual fusion surgery. It is non-surgical. It's very easy to administer in a treatment room, and it's done very much like a discogram. Instead of injecting dye, you inject the cell therapy. It is off the shelf, again, as a frozen product. So it really means no complex logistics for the patient. Our treatment is done in a treatment room with a C-arm. We inject into the disc percutaneously. No surgery intervention involved, and our post-treatment hypothesis is reduced pain and hopefully the normalization of disc tissue. We've had very good preclinical results to demonstrate the safety and bioactivity in IDCT. We've injured rabbit discs to mimic human degeneration, and as you can see, we've shown restoration of disc height and normalization of tissue architecture that's not seen in controls. We've seen no toxicity and no safety concerns, and we used all human cells in all of our various animal models that it consisted of New Zealand white rabbit, Göttinger pig, a beagle model that we did in Japan, and a athymic mouse model. We did this athymic mouse model at the request of the FDA from a tumor genicity perspective, and not only did we not show any tumors, but we also showed the ability to grow disc tissue. This had a pretty dramatic impact on our technology, in addition to the restoration of disc height and tissue normalization, to be able to see this automation of disc tissue through these cells in an athymic mouse muscle pouch. So, as we move forward in our regulatory pathway, we really are after a global footprint for our product, because obviously degenerative disc disease is a worldwide problem. We are in a phase 1/2 U.S. clinical trial. We're about a third of the way through, having passed our first safety gate. This is our clinical trial in the U.S. is a multi-centered with 14 sites to date, and it's a double-blind, placebo-controlled, multi-centered study. In Japan, we've just began our clinical trial there. Tokai University is our first clinical site, and we're recruiting 5 others as we get the IRBs approved. We hope to have our first enrollment in May. So we are very excited about our progress both in the U.S. and Japan, and with this in mind, especially in Japan, as we look forward to a possible conditional marketing approval, our commercial manufacturing is extremely important. And when we look at this whole commercial manufacturing, I'll talk about that in a second, but I also want to tell you about our pipeline. As you can see, with our IDCT for lumbar degenerative disc disease in both the U.S. and Japan, we are beginning some good preclinical work around cervical degenerative disc disease, and obviously post-fusion or adjacent level. We're also looking at some cell therapy technicalities around discectomy and how we would do that after a discectomy surgery has taken place. We're also looking at annular repair. We also have some very interesting technology we developed around articular cartilage around other parts of the body, and facet joint pain, and then obviously a second generation IDCT. So as we look to our possible conditional marketing approval in Japan and possible other accelerated opportunities, possibly in the U.S. and around the world, our CGMP manufacturing for human use becomes a critical part as we scale up and scale out and develop phase 3 and commercial CGMP. We are currently kind of in between as we scale up through our bioreactor development, and we've done this and taken all of our manufacturing in-house. We work very closely with CCRM of Toronto and GE, and they've done a tremendous job in helping us scale up and scale out as we've gone through our various stages of bioreactor development. And our in-house manufacturing capabilities and expertise continue to improve. As you know, this is a very difficult phase of what we do, and we're making very, very good progress in our development and our scale up and scale out. So this is a very interesting slide, and it really has two purposes. Originally, it was the prioritization of our global markets, and we did this and obviously came up with the U.S. and Japan, and these were the considerations were around market potential, the regulatory environment, the manufacturing and clinical acceleration, and we endeavored to embark on this to both the U.S. and Japan, and then to look to medium term to Europe and other parts of the world, and then also explore long-term investigational opportunities such as China, Brazil, South America. This has done it's really held us in good stead, and given the manufacturing of our cells here, not here, but in Salt Lake City, Utah, this also shows the ability of our cold chain logistics after we achieve the manufacturing scale-up. We've used cryoport extensively in this cold chain logistics through both proof of concepts in Japan and also for our clinical trial, and we very much appreciate that partnership. And we know that cold chain logistics is going to be a big part of all of our cell therapies moving forward, and we really are committed to going from our freezer to the clinical freezer in one step, and we believe the cold chain logistics is going to be a very, very big part of that, not only from a logistical standpoint, but also from a cost basis. So if you look at the market size of the opportunity, the disc repair market in the U.S. is projected at almost $16 billion. Outside the U.S., it's about 30. So it's a very, very sizable market, and it lends itself to the ability to help literally millions and millions of people, not only in the U.S. and Japan, but around the world. We really believe that we can have a very disruptive effect on this treatment continuum, especially as you look at the current DDD treatment pathway leading to a surgical procedure. And if you look at that, that extends over a 2, sometimes to 4-year period with cost in excess of $115,000 per episode. We believe that we can disrupt that with a 6 to 9 month continuum and do that at damn near 1/5 the cost. So we think we can have a dramatic impact, not only in helping people with degenerative disc disease with a possible curative option, but we also think we can disrupt this from a cost perspective in a major, major way, and also being non-surgical. So in conclusion, I believe we have a tremendous opportunity to literally help millions of people with what today is a pervasive disease. We are looking forward to the results of our clinical trial, both in the U.S. and Japan, and moving forward as we continue, hopefully, to commercialization. We are currently in a Series C fundraising. We've done a convertible note to that Series C, which is now oversubscribed, and I'll be glad to talk about that if anybody's interested from the financing standpoint after this talk or the rest of the day while I'm here. Thank you very much. I appreciate the opportunity.